In this last video on amines, we'll be talking briefly about amine heterocycles. We've talked about heterocycles several times throughout the year, and keep in mind heterocycles are cyclic compounds that contain some element other than carbon. So there are heterocycles that contain nitrogen or oxygen or sulfur or multiples of those elements or a combination of those elements. Here we have a number of amine heterocycles, and we have a couple examples like piperidine and pyrrolidine that are completely saturated, as well as a number of unsaturated compounds. Many of these are aromatic. We have single ring structures such as pyridine and pyrrole. We have bicyclics such as quinoline or isoquinoline. So both of these have a benzene-like ring and a pyridine-like ring, but the nitrogen is in a different orientation. And then we have a 6-5 combo. This is an indole. There exist heterocycles that have multiple nitrogens, such as a 5-membered ring with 2 nitrogens. This is imidazole. A 6-membered ring with 2 nitrogens. This is pyrimidine. And the 6-membered ring fused with a 5-membered ring with a total of 4 nitrogens. This particular one is called a purine. Some of these may look familiar because of how we've mentioned them earlier in the year, but many of them may look familiar because you may have seen them in your biology textbooks. This, for example, is quinine, so we can see here that it has a quinoline. This is an anti-malarial medication. This is tryptophan, which is an amino acid, and we can see here we have the indole group that is part of this amino acid. And this example is adenine, which is the nitrogen base of the adenosine nucleotide. We can see that this consists of a purine with an amine substitution. If we want to examine the reactivity and the biological importance of different amine heterocycles, it's helpful to think about some of the physical and chemical properties. For example, we can think of the pKa values for the conjugate acid of a number of different amine bases. So imidazole itself, the conjugate acid, has a pKa of 6.95. And looking at the imidazole moiety in the amino acid histidine, the conjugate acid has a pKa of 6. So this is relatively close to physiological pH, and so histidine is very important for proton transfer. There are various amino acids that have acid side chains, but at physiological pH they will exist as carboxylates, and there are amino acids that have amine side chains that will exist as ammonium ions. But histidine is really important for proton transfers because it's relatively comfortable either as a neutral or protonated species. So that means that histidine can effectively facilitate proton transfers that happen at enzyme active sites as they are catalyzing particular reactions. If we look at thiazole, this is similar to imidazole, but this nitrogen is replaced by a sulfur, and that has a significant impact on its properties. So the nitrogen in thiazole, its conjugate acid has a pKa of 2.44. So that means its conjugate acid is much stronger, which means this is going to be a weaker base than imidazole itself. Thiazole is a component of thiamine, which is also known as vitamin B1. Here we have again the structures of pyrrole and pyrrolidine, which I flipped over from a previous slide so that we could compare it with these, which are the electrostatic potential maps for pyrrole and pyrrolidine. So we had talked about these compounds separately, comparing basicity as we're thinking about the aromaticity of this ring. And what does that mean in terms of the lone pair of electrons on the nitrogen? Because they are delocalized throughout the aromatic ring, that makes them less available to act as a base, and therefore this is a weaker base than pyrrolidine. And here, looking at the electrostatic potential map, we can see another representation of this. So in an electrostatic potential map, red is negative and blue is positive. And so in pyrrolidine, we can see a lot of negative character right around the nitrogen. And so we know that we have a lone pair on here, and nitrogen is more electronegative than the neighboring carbons and the hydrogen, but we can see that this negative character is localized right here. In the case of pyrrole, that red, so the negative character, is really distributed throughout the ring, which leaves the nitrogen without a lot of electron density, which makes it a lot less basic. Finally, we can consider some of the reactions of amine heterocycles. Earlier this semester, when we were talking about electrophilic aromatic substitution, we mentioned how these reactions can occur on compounds other than benzene and substituted benzene, but how they can also occur on heterocycles such as pyridine or furan. So will the reaction occur? In many cases, yes. Where will the substitution occur? This is something that can be determined experimentally. You could carry out this reaction, isolate and characterize your products, and find out whether nitration occurred at this position, or this position. In the case of furan, you could carry out this reaction and characterize the product, determine whether in this case, sulfonation occurred here or here. Or you could use mechanistic information to predict the position of substitution. So for example, if you considered whether these electrons are shared with the electrophile, what does the intermediate look like? How is that stabilized by resonance? You can compare that to the situation if the reaction occurred at this position. Draw the intermediate, think about resonance structures, and also if reaction occurred from this position, draw the intermediate, think about the resonance structures. So by thinking about it mechanistically, you could predict the location of substitution, and then it could potentially be verified experimentally. So I'm not asking you to memorize this information, but I just want you to recognize that you do have the tools to be able to figure it out. Let's just look briefly at a couple of examples of electrophilic aromatic substitution on amine heterocycles. So pyrrole can undergo bromination at this position selectively, and the reaction conditions are pretty mild. What we saw previously for benzene-like compounds, we needed to use bromine and a Lewis acid catalyst to be able to get the reaction to occur. But here, simply using molecular bromine, and even at low temperature, this reaction works pretty well. In comparison, the reaction of pyridine occurs at position 3, so further away from the nitrogen than what occurred here, and this requires much more forcing conditions as well. Here we have a comparison of the bromination of two bicyclic heterocycles, quinoline and isoquinoline. And in the case of bromination of quinoline, the substitution occurs at this position and this position. So notice it occurs on the benzene ring, not on the pyridine-like ring, which shouldn't be so surprising since we just saw that pyridine is not extremely reactive. When we look at isoquinoline, where the nitrogen is in a different position, the substitution again occurs on the benzene-like ring, the same relative positions, but we do have a significant difference in regiochemistry. Whereas in the case of quinoline, we get substitution here compared to here at almost a 50-50 ratio, whereas isoquinoline, we get substitution here compared to here at a 90-to-10 ratio. So a lot of this work has been done experimentally, or it could be done experimentally, or you could use your mechanistic understanding of electrophilic aromatic substitution reactions to be able to predict where and how likely a reaction would occur. And this concludes our brief look at heterocycles. Keep in mind, again, some of the physical properties as well as some of the chemical properties are similar to other types of amines, and some of them are unique to amine heterocycles. And this is important to consider because of their relevance in many biological molecules and therefore their importance in many biological reactions.